Hello everyone, myself is Dr. Satish Maurya and in the current module we are going to discuss about root development. In the next three modules we will study how the various tissues in a seedlings are derived from meristems. Shoot apical meristem, also known as a SAM, are responsible for the development of stems, leaves and whereas root apical meristem, also known as a RAM, is responsible for the growth of a root or underground network. In this module, we shall see how the radicle that emerges from the seed coat during germination develops into the root system of the plants. So as we all know, underground, if you'll uproot any plant, you'll get a network like huge network of a root. So there will be primary root and secondary roots. That's what we are going to discuss in this current slide. Current module is divided into three different objectives. So the coming to the first objective, which is nothing but development of the primary root and root cap, whereas the second objective talks about the development of root hairs, lateral roots and secondary growth in roots. And the third objective of this module is sensing of gravity, light and moisture by the roots. Coming to the first objective, which is development of the primary root, where upon germination, the radicle emerges from the seed and develops into the primary root called the tap. In dicot plants, the taproot persists of and gives rise to lateral roots that emerges from the primary root axis in an acropetal direction. Whereas in monocot plants, the initial taproot soon produces lateral roots that equal or exceeds the primary root in size. Hence, there is no well-defined single taproot. As seen in the earlier modules, the root apical meristem consists a group of slowly dividing cells in the quiescent center which is mitotically inactive and functions as the organizer of the root stem shell niche. The meristematic zone lies immediately above the root cap and consists of rapidly dividing pluripotent cells originating from the quiescent center. The transition zone lies above the meristematic zone, which is highlighted here, and develop competence for elongation near the elongation zone which lies above it, so which is highlighted here. The cells in the elongation zone elongate rapidly and show the presence of large central vacuoles. Root hairs initiate from epidermal cells in this zone. The differentiation zone lies above the elongation zone, which was already highlighted. Here cells do not elongate further, but the root hairs show rapid tip growth. Lateral roots arise in the region above this zone. Oxygen entering the root from the shoot is transported through the still tissue towards the root tip. So as you can see here, from the tip or upper part, the oxygen is transferred to the tip of the root. At the root tip, this auxin along with the auxin synthesized by the root apical meristem is redistributed upwards through the lateral root cap and epidermis. Subsequently, auxin is refluxed through the stele back towards the root tip. During transport, auxin maxima are established at the root tip and in the region above the differentiation zone. Lateral roots develop from the latter, so which is highlighted here, and mutants with defects in the auxin transport and signaling like plethora, pinoid and monopteros, which we have seen in the earlier modules, show premature arrest of the root meristem and root stem cell function. Coming to the radial development of the primary root. So radial development is nothing but the TS of a root. So when we talk about the TS of the root, on the radial axis, the primary root consists of concentrically arranged stem cells in which the distal cell divide and finally differentiate radially from inside to outside to form the vascular cells The outermost layer being the pericycle, which is highlighted here in the green color. Then followed by that, the endodermis, and after endodermis, there is a presence of a single layer cortex cells and epidermal cells. In Arabidopsis, the typical root structure consists of a single layers of epidermis, cortex, endodermis, and the pericycle. The vascular structure shows an exarch xylem where metaxylem is directed towards the center and the protoxylem is directed outwards or to the periphery. However, the xylem and phloem shows an alternate arrangement on the radial axis. As seen in the earlier modules, the transcription factor, that is short root or SHR, plays an important role in regulating the differentiation of the pericycle and vascular bundle, which is highlighted here, from the initial stem cell. Further, in addition, movement of short root protein to the ground tissue activates the scarecrow gene, that is SCR, which regulates the differentiation of the endodermis and the cortex in the ground tissue. SHR or scarecrow mutant shows the distortion along the radial axis and lacks the endodermis. We shall now see how the patterning of vascular tissue takes place in the developing root, or the another objective of this module, where in the developing primary root, auxin response is highest in the xylem axis, while cytokinin response is highest in the adjacent cell or the nearby cells. Auxin signaling induces expression of an inhibitor of cytokinin signaling, the histidine phosphotransferase protein 6, that is AHP, which is highlighted here. So AHP6 at the marginal position of this axis, particular xylem. Auxin also induces the transcription factor target of the monopterose 5, that is TMO5, which forms a heterodimer with the another protein, which is a lonsome highway, that is LHW, to activate the expression of cytokinin biosynthesis gene, that is lonely guy 3 or LOG3, where the cytokinin moves to the procambium cells and the phloem. And as you can see, in the procambial cells, cytokinin levels are limited by the activity of cytokinin oxidase CKX3. A cytokinin maxima occurs in the region adjacent to the procambium, which is highlighted here, which specifies the differentiation of phloem elements from the procambial cell. The activity of HDZIP3 transcription factors play an important role in non-vascular patterning. We have already seen that the HD-ZIP3 transcription factors like the Arabidopsis thaliana homeobox 8 or ATHB8 regulates patterning of central region, and the HD-ZIP3, that is fabulosa, patterns the adaxial regions during the early embryogenesis. The activity domains of HD-ZIP3 transcription factors in the roots are regulated through the activity of microRNA 165, where microRNA 165 moves from its site of synthesis in the endodermis to restrict the SD-ZIP3 transcription factor, where it is showing in the figure, to the central stem. The short root, that is SHR protein, which is expressed in the stele region, diffuses to the endodermis, where it dimerizes with the scarecrow protein to endorse the expression of miRNA 165, as you can see here. The miRNA or microRNA diffuses into the stele and degrades the mRNA of SD-ZIP3 transcription factors, causing the gradient of these transcription factors with the maxima in the center. The gradient specifies protoxylem and metaxylem with highest level of SD-ZIP3 activity promoting the metaxylem and lowest level promoting the protoxylem cell identity. Hence, several signaling components that are active in the development of the vascular tissue in the embryo are expressed throughout root growth and development. Coming to the root cap development, which is an important feature of the primary root, which protects the ram, that is uh, root apical meristem, from abrasive damage during the growth through the soil. The root cap is formed from the columella stem cells lying distal to the quiescent center, which divide asymmetrically to generate one stem cell and a differentiated root cap cell. The lateral root cap originates from initials that are located laterally to the columella stem cells and extends upward to the end of ram. These cells divide to form the epidermal cells and lateral root cap cells. Two transcription factors, Fez and sombrero, that is SMB, are required for correct execution of the root cap developmental program and are active in root cap stem cells and their immediate daughters, which are highlighted here in yellow color. These transcription factors are antagonistically to control periclinal divisions in the epidermis, lateral root cap stem cells, and columella stem cells to form the lateral and columella root cap in the young root, so which is highlighted here. The terminal differentiation of the columella cell is brought about by transcription factors called the BSkin, that is BRN, which are induced by the sombrero transcription factor. Further, the root cap maintains a constant size by production of new cells from root cap stem cells and detachment of cells from the outermost layer of the root cap by programmed cell death, which is highlighted here, where the programmed cell death or how the programmed cell death happens, which is highlighted in the figure. The detached cells are called border cells, which are embedded in mucilage that is secreted by the last row of the columella cells. So columella cells do produce the mucilages. And the BRN transcription factors also plays an important role in mucilage accumulation and their secretion in the roots. Moving to the next part of this module, which is the root hair development, which is an important feature of the root, which are the site of water and nutrient uptake from the soil. In legumes, they are also the regions for microbe interaction that lead to the formation of nitrogen fixing nodules. Root hairs are the extension of the root epidermal cell, which are highlighted here in the form of a projections, where the outer epidermis is composed of two cell types, those that form root hairs, that is known as a trichoblast, and those that don't are known as a non-trichoblast. Trichoblasts are seen to lie above the junction of two cortical cells, and where those in contact with cortical cells are known as a non-trichoblast, so which are highlighted here. So this is cortical cells, and if they are present between the two cortical cells are known as a trichoblast, and if they are present just above the cortical cells are known as a non-trichoblast. The formation of trichoblast seems to be in response signals from the underlying cortical cells in the root hypocotyle junctions. However, every epidermal cells form a root hair-like extension during the early seedling growth, and probably due to the multiple layer of a cortex cells seen in the hypocotyle regions. The root hair cell fate represents the default fate for a root epidermal cell. Transcription factor, or known as TTG, or also known as a transparent testa glabra, which is highlighted here in bluish color, and it, along with its interaction with the other transcription factors in the epidermal cells, decides the formation of root hair or it can inhibit the formation of a root hair. So ultimately, it decides the formation of a trichoblast and non-trichoblast cells and their fate. So as we can see here, the moment it interacts with the different type of transcription factors like WER and CPC, they can regulate the form or interact or regulate the expression of a GL2, that is glabra2 protein, or also they can inhibit the GL2 protein. So if in case the GL2 protein is inhibited, ultimately it will form the root hair, and those type of cells are known as the trichoblast cells, and they will ultimately form a root hair. However, if the TTG is reacting with or interacting with the WER protein, ultimately it is going to regulate the formation of a glabra2 protein, and the glabra2 protein is known to inhibit the formation of a root hair. So as we can see here, the TTG along with the CPC will regulate the formation of a root hair, whereas TTG along with the WER is known to the function for the inhibition of the root hair. So here it is highlighted in detail. Besides CPC and WER, TTG is also known to interact with another basic helix loop helix type of transcription factor called GLABRA3, that is also known as GL3, in regulating the GL2 expression. Interestingly, some of these transcription factors are also known to play a role in the development of trichomes in leaves, which we shall see later. Once specified, the root hair cell wall begins to bulge out and elongate. Elongation of root hairs depends on polarized cell expansion and exocytosis of vesicles containing cargo molecules such as cell wall polysaccharides and cell wall glycoproteins. These vesicles are incorporated into the newly forming cells or cell wall at the tip of the root hair. Root hair growth is rapid and occur at the rate of 1 micrometer per minute. That's quite fast. In the dicot taproot system, the embryonic root develops into a persistent primary root, along with which new lateral roots are formed, which are highlighted in the figure. These in turn develop the lateral roots, leading to a highly branched root system. Auxin plays a major role in the initiation and growth of lateral roots. A basipetal auxin concentration gradient with high level of auxin in the root tip controls the initiation of lateral root primordia. Auxin transport from the SAM to the root contributes to the subsequent growth and emergence of the lateral roots. The auxin maxima is maintained through the pin protein and signaling by the AUX or AUX and IAA and ARF transcription factors. The lateral root primordium arises from the pericycle cells, which is highlighted here in the green color, that have retained their stem cell function and even after moving from the ram. So even after the pericycle cell moves away from the ram, it retained its function or stem cell function. In response to the auxin maxima in this region, these stem cells show asymmetric anticlinal divisions forming a single layered primordium of 8 to 10 cells. So as you can see here, in auxin transport and signaling mediated cell division, where the anticlinal and periclinal divisions takes place to grow the lateral roots. Further, the quiescent cells are formed or demarcated, and the subsequent growth take place in the same manner as seen in the primary root development. Further, the plasmodesmata connections between the lateral root cells and the primary root cells are lost, and these cells are then pushed apart by the emerging lateral roots. The lateral root is symplastically isolated from the vascular system of the main root till vascular tissue differentiation in the provascular tissues of the lateral root leads to the establishment of the connection to the main vasculature. Further, moving to the secondary growth in the roots, in many species, secondary growth take place in root and result in thickening of the root diameter by the addition of vascular tissues. Initiation of secondary growth generally occurs in the differentiation zone. The residual vascular stem cell and the pericycle cells, which are highlighted here with the red dotted lines or arrows, are divided periclinally. The root vascular cambium activity occurs in response to the auxin and cytokinin signals received from the shoot. So as you can see here, which is the TS of a root, which shows the secondary growth, where the vascular cambium forms a continuous ring around the primary xylem and it divides periclinally to give rise to the daughter cells that differentiate into the secondary xylem, which is highlighted here, towards the inner side, and secondary phloem cells towards the outer side of the root. Some of the secondary xylem cells differentiate to form medullary rays, which plays an important role in radial transport within the root system. So, the roots also form an outer protective layer called the periderm, which also originates from the pericycle. The pericycle divides periclinally again to form the cork cambium, which is highlighted here. The cockambium divides and differentiate to form the subraised cork cells towards the outside, which are dead at maturity. So ultimately, they act as a protectors, and the phylloderm, which consists of a living cells on the inner side. The secondary growth has functional significance with regards to soil resources acquisition and transport, as well as in interaction with the soil organisms. Besides anchorage and the acquisition of water and nutrients from the plant, roots show other functions that are also important for the plants. Roots show positive geotropism, negative phototropism, and positive hydrotropism. All these tropic responses are based on the environment sensed by the root tip and lead to altered direction of growth in the elongation zone of the root. The phototropic response is mediated by the perception of a blue light by the photoreceptor phototropin, that is FOT1 protein. The FOT1 protein is expressed in the cortical cells of the root, which is highlighted here, and the cortical cells of the root elongation zone, and its expression is higher at the shallow soil depth where the blue light penetration is the greatest. So in response to blue light, the FOT1 is expressed. The gravity is perceived by the cells called the statocytes, which contain starch-filled amyloplast known as the statoliths. So as you can see here, the statocytes followed by the amyloplast, which are nothing but the starch filled within the cell, which are highlighted with the black small dots. The statocytes alter the localization of a components of the oxygen transport machinery, that is pin proteins, to the lower plasma membrane domain of the gravity stimulated columella root cap cells. This causes oxygen to relocate to the bottom side of the root tip, acting as a trigger for the positive geotropic growth or root growth. However, the mechanism by which roots display hydrotropism is not so well understood. The moisture gradient and the gravity force are both detected in the root tip, and they both triggered a growth response in the elongation zone, which is highlighted here. However, the all such responses are triggered or mediated by the auxin flux. Hence, auxin is the long-range signal that translates gravity or water signals into a growth response in the root elongation zone. We have seen this module how a small number of a stem cell at the tip of root generate all of the cell type through divisions followed by the cell differentiation and regulated cell expression to form a root system in the plant. So as I earlier said, like start of this particular module, that we have seen the network like structure under the ground of each plant. So now we know how they are developed. Because root growth is indeterminate, these processes are continuous, resulting in all developmental stages being present in all at all times. In the next module, we will study the development of stem, which conducts the water and nutrients taken up by the root to the aerial region, and that is again the interesting topic. So, see you in the next module. Thank you.